Hi, welcome everyone to this morning's session. Thank you so much for taking the time out of your busy schedules to come and learn. So today's topic will be controlling microbial growth by disinfestation and sanitization, which will be presented by our speaker, Dr. Leow. But before we begin, I'd like to just lay down some ground rules for everyone. If you have any questions, please use the Q&A feature within Zoom, and in order to put your questions in. The reason being is because we want to be able to track all the questions so that all your queries can be answered by our speaker today. Right. Okay, so it's now 1:10. Without further ado, we have a lot to cover today because it's a very pretty heavy topic. So I'd like to pass the time now to Dr. Leow to maybe introduce himself a little bit, and then we can get started on today's session. Okay, Dr. Leow, over to you.
All right, thank you very much, Chris, and good morning to all the attendees. My name is Yee, so I'm currently, one second, let me share the slide with you guys first. Hang on. Oh. So, okay, here we go. Chris, can you see the slide? Okay, cool. Yeah, it's working. Yeah, good morning. My name is I'm Dr. Leow, or just call me Yee alone. So I'm currently a lecturer in Universiti Sains Malaysia. So before I would like to share with you about what the disinfectant and sanitization and how are we going to use the basic, the kind of the fundamentals knowledge that we have to use them. We need to understand about how are we going to control the microbial growth. This microbial could be involving the bacteria, viruses, fungus, or whatever. So, but today we know that because recently is a pandemic season, right, dominated by this COVID-19. But today, so I will try to focus more on some viruses effect. So before I start, I would like to thanks to the UB Corporate Solutions for providing me this platform to share my today's talk with you guys. So there are a couple of different highlights I'm going to touch on today. The first one, of course, I will touch on is we have a kind of like brief introduction to pathogens and microbial. And the second topic that I will touch on today is difference between the sanitizers and disinfectants that we are going to use commonly in our daily life. And what kind of the chemical disinfectant groups that also will be discussed. And before the end, also we're going to have some brief idea about how are we going to test the effectiveness of the disinfectant that we use in the market today. So last but not least, then we will finish up with the disinfectants for general disinfections for the use of against the environmental and in-house surfaces. All right, without further ado, so now I'm going to try to share with you about what is the microbiology first. So of course, when we talk about the viruses, no matter viruses, or bacteria, fungus, all these things, they are grouped under the topic of the microbiology, like the word of micro, which means something small life size. Biology itself means life thing. All right. So what's the microbiology? Is we're going to study the microorganisms to understand about the unicellular, which means they're only single cell, or they are grouped under like a cell cluster microscopic organism, which means probably like for the bacteria, they consider as a unicellular, but for the parasite, they consider as a multicellular organisms. So they also include some like eukaryote stuff for the microbes things, for example, the fungus and protists. All right, I will show you some photos or images on this later. So which means that when we talk about eukaryotes, unlike the unicellular eukaryote, it has all the cells they form with a nucleus. All right. So when we talk about the prokaryotes, for example, like bacteria, which means that some of the cells it may form with a single cell, but it doesn't have the particular nucleus with them. For example, the bacteria and the viruses. Viruses usually they are not classed as a living organisms when they appear in the environment because they need a host to run their mechanism. All right. So the bacteria, as we know about this, they are the single cell, for example, unicellular, they also consider as we call it as a prokaryote because the genetic material are not enclosed within the nuclear membrane. All right. So usually what you can see in the on the images over here, these are all the different kinds of the bacteria. Usually they come in the group form, which means that they multiply themselves by dividing one to the other, or we call it as a fission binary. So which means that there are a couple of different different forms of the bacteria. Like for example, we know that certain bacteria, they know as a cocci. For example, like streptococci is a most famous one. This is usually, we call it as a like human infection. Also like bacillus, which means the shape itself look like an oval shape. So like bacillus as well, they belongs to this kind of the group of the bacteria. And the other type of the bacteria is like a Spirulina. Spirulina, this is we know that leptospira, which found from the we call it as a found in the wild mouse, the wild rodents or the wild mouse, which mean the mice, which mean that if let's say any rat they're contaminated out there on the street or whatever in the wild area, so they most likely they are contaminated with they are the vector to the leptospira. So leptospira considered as one of the very serious disease for the human, because this one usually cause a very severe, or it can lead to death if let's say they are not treated in the early time. So this is a couple of the different examples that I would like to share with you guys. This means because all the bacteria, they can be known from their shape, and then they call it according to their name. For example, diplo, which means double. Staphylo, which means they are considered as a 3 or 4. But if let's say more than 3 or 4, they're going to call it as a strepto, and more, we know it as a cluster. So sometimes you'll be noticed with a lot of the different bacteria like Staphylococcus or Streptococcus, so just because of they have their shape and the number of the bacteria, how they must one to each other. All right. So another type of the microbes that we need to understand is we call it archaea. Archaea usually it doesn't belong to the kind of pathogens, but archaea more relevant to as known as environmental bacteria or environmental organisms. They are not considered as a bacteria. To be honest, archaea was known as one of the like ancestor to more of the living organism, because all these archaea can be found in the extreme environment. For example, in the spring water or in the very cold temperature, right? So these archaea usually like Antarctica or in the near to the spring water, near to the lava site. So you may see that some archaea can be found or living there. All right. So this because belongs to the ancestor to most of the microorganisms, but nobody found that yet is belong to any of the pathogens so far. All right. So another type of the microbes that we need to understand is we know it as a fungi of some type. If let's say singular is fungus, for plural is fungi. So fungi that we know here is usually we know is a mushroom that we know that we eat a lot from the mushroom, right? So this is all the mushroom that we can see, like that you can see in the markets. All right, like this mushroom, because it has a fruiting body over here. But the important point is what we see from our naked eyes is their fruiting body, which means the mature part of that. However, when if let's say we try to look into the details, they have lots of mycelium grow between their parasitic substance. For example, this dead wood, so they have a lot of mycelium. So this is a mycelium that causes a problem. Let me show you something about this. Okay, let's see. These are all the we call aspergillus. All right, this aspergillus, of course, when you try to look into through the naked eyes, they look something like the black color near to the wall. But this aspergillus, when because they form through the spores, right? They can form a lot of spores. So when these spores they start to they started to hatch from the fruiting bodies, then they will be circulating throughout the air. So all these spores, when we try to culture in the lab, you may see that they start to form into the fruiting bodies. But the problem is the spore itself might be allergenic to the human. That's why this is sometimes it can cause a toxic as well. For example, aspergillus and penicillin, they are two of the big problem for a lot of the indoor environment, especially in the hospital. If let's say they have a certain exposure to the humid or something cold, because they don't need any lights to grow, not like other plants, so they can grow easily in the confined area. So this is will cause our esophagus the bleeding problem. All right, because it's due to the allergy. Yeah. So another type of the microorganisms here I would like to introduce is we know as a protozoa. I think this protozoa we can find easily from the river or from any part of the water. But for example, amoeba itself amoeba has so many types, but this is a family we call it amoeba. And the other type is like a shoe shape, which we call it a paramecium. So all these two, they start compared to the microbes organism, it still belongs to it doesn't belong to the microbes part is a unicellular, although it is because they have their own mechanism for them to make their own energy or to demand and to look for food. So their size is still much bigger compared to the bacteria. All right. So this amoeba sometimes we can find, or we call it protista, is like giardia. Usually that cause to our human. If let's say we have some food contamination, the giardia we get, so it leads to the food poisoning. All right. So another part I think most that we know is like in our small children, the school children now, most of the school, like since the WHO, they will enter all the school children, they'll be introduced to the albendazole, one kind of the drugs to kill the intestinal parasite. So another part, the parasite also considered as part of them, like for example, flatworm. There are so many different worms. They have the trematose or the liver flukes. They have the flatworms. They have some roundworms as well. For example, the current project I work with the Thailand collaboration is we work together. Is we call it as a liver worms or liver flukes. It's a kind of the worms that are introduced by eating the raw fish. All right, from the freshwater raw fish, and because they have the larva found in the raw fish, so when the people they feed on this uncooked raw fish, so they might the larva may start to develop and turn into the mature worms and start to reside within their liver, so leads to the liver cancer. All right, but I'm not going to touch too much on that. I just say that this is a problem of the worms, because the problem is not the worms itself. It's about a lot of things that we cannot see through our naked eyes. For example, when we eat the uncooked material from the, say for example, raw fish or certain kind of the freshwater milk product, which are uncooked. So we are introducing by eating their eggs into to us. So when the eggs of the parasites come into our body, then they start to hatch because the environment suits them. They start to hatch, turn into the larva, and the larva will start to flow according to our within our circulating system, the blood system, and then they will find a way. Usually they will stick themselves within the liver, because liver to them, they have a lot of nutrition part over there. All right. So now, last but not least, I want to introduce to you all today, our focus is more on the viruses. So viruses different from other microbial groups. The viruses itself is very small, until usually we call it as a nanometer. We cannot see it through our naked eyes. Of course, even the normal microscope, we also cannot see them. We have to use a kind of special microscope. We call it as an electron microscope, which means that the magnification rate is much strong and much higher compared to the normal microscope. And it belongs to I can't say it's an organism. It belongs to a thing that it doesn't have the cellular. All right, because they form all this cellular, or they need all these things when they come into the host. When they appear itself, actually it belongs to a single strain of like genetic material. It doesn't have anything covered there. I will show you some photo later. And of course, like I mentioned just now, the viruses itself, they reproduce through using the cellular machinery of other organisms. All right. Of course, they belong to most of them belong to the parasite, which means that it's not the parasite I mentioned just now, not the worms, but which means that when we talk about parasite, which means that they cannot do their own contribution, but they need to rely on the host to get their benefit. The host cannot get any benefit from them. So, but the virus need to get some benefits from the host, which means that this one we call as a parasitic infection. If let's say two of them, they're going to get the similar benefits, so we call it as a symbiotic interaction, which means that they're not going to hurt each other. However, they will get the benefit from each other. All right. For example, the T4 bacteriophage and adenovirus, all these things. And then the genetic material is very simple. It's exactly like what we have in our human as well, or in the bacteria and all living organisms. It has a nucleic acid, or either in DNA, or for our today the SARS-CoV-2, or at least to the COVID-19 is a RNA viruses, and it has a normal protein code to form their capsid only. So that's it. This is so simple for them. All right. So these are some different shapes of viruses that we found in the environment, or that we know currently. For example, we know it's a tobacco mosaic virus. You may see the shape is like a, it is differently. All this because of the after they identify or classify through the use of electron microscope, so they come up with a diagram. And this is a T4 bacteriophage. As you see the bacteriophage, the name phage itself is a virus, bacteria, which means this virus actually is currently been engineered, used to infect the bacteria as well. So which means that in our laboratory, we use a lot of this kind of the bacteriophage. Now, for example, this bacteriophage, this is a bacteria. They infect the bacteria to try to, we try to do a lot of like protein engineering work in the laboratory. All right. So what it did is they have the genetic material on their head. We call it a head over here. It has a tail, the end tail here. So they will inject the genetic material through the membrane of the bacteria into the system. So once the genetic material introduced into the bacteria, so we'll take over all this kind of the host genome system to try to facilitate the bacteria to replicate their DNA, and at the same time to make some protein to form their capsid again. So more and more will form within the bacteria, and once the time is there, then the whole cell will rupture, and more big, I call it a virus baby, will form and go to infect to another new cells again. So this is another kind of like HIV viruses. This is Rausi. This is the shape itself is pretty much similar to the SARS-CoV-2, but of course SARS-CoV-2 have their different shape. I'll show you later. So it has the, like for example, is let's say we look into that, their form is very simple. They have their genetic material within their core, and they have their out within their layer, they have the capsid, and some of them they call it as an envelope virus, which means that they have another lipid envelope outer of their layer. All right, so this the form itself is very simple. However, their interaction with the host, the receptor, is very specific. That's why they can infect the host or the human or whatever animals when if let's say the interaction, the receptor and the ligands of their lipid are compatible, right? So they can attach it tightly and then start to introduce the material, like I will show you. They start to introduce the material into the living organisms. All right. So another part is I would like to introduce is we call it prions. I think we will see a lot of these words later on when you use a lot of like disinfectants. You will say that whether they can be used against a prion or virus. So this prion itself is pretty much similar to the viruses form, but however it doesn't have it's a very kind of like proteins. Instead of like being as a RNA or DNA, it's a very simple protein that believed to cause the disease, which means that some protein fragment in the environment that that we can't see them. So probably when they come into the host, then they start to create the problem again. Like for example, certain, but of course I cannot group the allergic problem into the prions because prions is a different form. All right. Okay, another material which is pretty much similar to the prions is because of virus. The virus is a small RNA which has no associated with protein that causes disease in the plants. So this is the virus itself usually found in the plants. All right, but this is very rare in the environment. Don't worry. I think the virus itself still need to be concerned but I need to introduce all these kind of terminologies just because of before we want to know more about how to use the proper use the disinfectant, what can it be used against. So we need to understand, because sometimes the MSDS will show you their effectiveness, right? So we need to know about these terminologies first. All right. So I would like to share some history about the microbes. Of course, all these microbes has been in the earth since the earth is formed billion years ago. However, after so much of years ago, then a lot of, because many years ago, we found that humans keep infected with the disease. Nobody know how come the disease is come from. But in the old times, there was some ruling to say that the disease might be because from the curse, right? People curse from it, and that's why there are a lot of different kind of practice by human beings until to the late or to the 19th century. There was a physician named Louis Pasteur. Of course, when you look at the Pasteur, this is quite famous because all the milk that we drink today, we are going through all this milk has been sent through the process because of pasteurization, because of the names of Pasteur, which means that he was the first guy to found that to find that all the organisms are present in non-living organisms, for example, on the solids, liquid, and in the air. Because before him, nobody knows what is a microbes until he found that there were some microorganisms in the environment. So which means that the microbial life can be destroyed by the heat. He proved that once we use the heat, we can try to do some sterilization. That's why he's the one who come up with the word of the aseptic techniques. That's why all these aseptic techniques until today is still practiced in the hospital settings. All right, because of this Louis Pasteur, he's the one who come up with all this. But because of him, today we managed to use a lot of different disinfectant and sanitization because of he introduced this kind of aseptic techniques against the living microorganisms. All right, so because of this aseptic technique, as I mentioned, has been used widely in the laboratory, in our today medical setting as well, to prevent for the contamination of the environment by the microorganisms. All right, so what he shows in the past was he did a very simple experiment. He prepared two different broths. This is a broth or the soup. Then he has to boil them together, but he set up two different experiments. One is after boiling, he wait and then he make something like this all happen in the glass. This is a glass tube. So he leave it there for years. Actually, this is what I was told. A friend of mine told me the product itself was still kept in French, the Louis Pasteur lab, Louis Pasteur Institute in French, to show that this is the first experiment that shows that the broth itself is still not contaminated until for so many years. But I'm not sure because I never seen the true case. All right, so which means that he shows that after boiling the broth, so nothing happened within the broth, within the soup, nothing grow. As long as you have to keep everything in the sterile format, which one this is a closed end tube, closed end tube, which means that the end tube it doesn't have any exposed to the air. Another experiment setting, he shows that after same thing, boiling, however, he leave the lid open, leave it in the air for a while, and then he found that the microbes start to grow within the soup after a couple of months. Of course, for us today, we found that this experiment is so simple, it's nothing. However, during that time, the microscope was not found yet. However, this is a big experiment because nobody knows what's going on with it. However, he tried to discover that this is all caused by the contamination of the microbial in the air. All right. After him, another guy is a Robert Koch. So thanks to Robert Koch and thanks to Louis Pasteur and Robert Koch and some of that, because these are the two important pioneers who found out that all the disease that we have today. Because Louis Pasteur found out that environment has a lot of microorganisms. However, Robert Koch, he found that microorganisms itself are the one that cause a lot of diseases in the human. So what he did in his experiment was, he found that when in the old time in the European country, of course, there are a lot of the dead mouse found on the streets. So he get a dead mouse and he brought it to his own laboratory in his lab. Then using a very normal during that time, because this is near to the end, early of 20th century, the microscope has been invented. Then he found that he stained the blood of the dead mouse. There are some difference or some certain kind of tiny organisms found within the dead mouse. Then what he did was he tried to use the dead mouse and culture them on the plate. Then he used one of this single colony and introduced to a healthy mouse. Let's say this experimental mouse. And then once the healthy mouse itself died after a couple of days. And he took out the blood again and to look under the microscope, and he found that these two microorganisms looks identical to each other. So since that time, Koch come up with the hypothesis saying that he's finding. He said all these things because they think that because of the infectious, and this infection is caused by the microbial infectious disease. So he's the first one who discovered all these kind of like the microbial infections. And later on, people start to have better technology. Then we found that a lot of diseases not only caused by the bacteria, but it can be caused by viruses and even the fungus as well. All right. All right. So now, because since we know how are we all this history, some background, right? So now we need to know about the pathogens. Of course, in the environment, we have more than billions. I can say more than billions of the microbial things is there. However, not all of them are fully characterized yet. For example, but out of them, only like maybe 0.2% or close to 1,400 known species of human pathogens so far we know. they are made up of like viruses, bacteria, fungus, protozoa, or helminths, which means it's worms. So most environmental microbes are not harmful to human. We need to know. That's why, of course, during pandemic, we need to be aware. We have to keep everything clean. But if let's say the pandemic is over, of course, we still need to keep the hygiene, but we cannot kill all the organisms in the environment. The first thing is because the organisms itself, as I said, most like 99% of them, they are required in the environment just for the balance, the ecosystem balance, because of course they need to be found in the soil, that we call it as the nitrogen fixation bacteria to help the plant to grow. Then certain kind of the plant, they also need the viruses for them to do some symbiotic process. And also we need the microbes for our, you know, in our tummy. I think that there's a reason why sometimes we need to, the doctor will prescribe some probiotic to our small children, right? So this probiotic actually, they help our body, we call it the microbiome, to grow much better. The number of the microbiomes found in our stomach is equivalent to the number of cells within our human. Is about like how more than millions of the microbiomes that we can find in the back. So but during pandemic, we have to be aware. But of course, we cannot drink something like to kill all the bacteria for ourselves. But we need to be aware with our environmental microbes only. All right. So this is I really need to touch on with this. So because it doesn't like a lot. The advertisement, they like to highlight saying that it can kill 99% of the bacteria. So it gives a kind of like assumption that people will think that all the microbes are bad. It doesn't seem like that. Only the pathogen itself are bad. So, but not all the microbes are bad to us. All right. But of course, we hardly to identify through our naked eyes which one is good or bad. So during pandemic, we just need to be aware to keep our cell hygiene only. All right. Pathogenic microbes account, like I say, less than 1% of the total number in the microbial species on the planet. So this is like certain, just now I tried to highlight. So this is the image to show you what's the difference between the cellular or the living and non-cellular. Cellular cells, which is come from the word of the cell, which means either the living and non-living. So most of the things like the parasites, protozoa, fungi, prokaryote like bacteria, they consider as a cellular or living organism. How can we group them? And because first thing, they know how to feed themselves. Second thing, they can make themselves the energy, the ATP within themselves. They don't need to rely on others. So because of that, they can get the food and they can start to look for their either for the well organism like worms, they can look for their partners. And for normal like this, they can start to do their binary fission without the help of others. Compared to the viruses, they consider as a non-living. Why? Because of course they only contain the genetic material in their core area. And all this part protein area, they make it from the host. They need to use the host machinery for them to heal up, to build all this. Which means in another way, if let's say they cannot find the host, like for example, today we put on the mask, right? So they cannot come into our system, which means they will be dying easily in the air. They are quite weak in the air, all right? Like pioneer as well, this is a protein, which means, so this is difference between the cellular and cellular organisms. All right. Just now we already understood about how all the differences between the living organisms, right? So now I would like to introduce you the difference in terms of sizes of all these kind of the organisms. Of course, we know that atoms belongs to the smallest particle, then followed by the DNA. And then usually all these viruses, all types of viruses, they fall within this category from 10 nanometer to 100 nanometer. It's very small, impossible we can see through our naked eyes. Even though we put the glass on, we cannot see them. And then the bacteria itself is about like 100 times much bigger than the viruses, 100 to 1,000 times, depending on what kind of bacteria and the viruses. So bacteria, the size itself is roughly from is around 1 micrometer until to maybe like 20 micrometer, not 20, maybe 10 micrometer, only depends on the bacteria. No, until to 2 micrometer, not 20, sorry, 2 micrometer. So, and then followed by is a red blood cells they found. So for this one, we know that now we can see the difference. So that's the reason why red blood cells, the size itself is so big. A lot of the bacteria or even viruses, they will use our cells as their living organism, their living place to live on, to get the resources for them to keep the live on. So our human cells is much bigger now. For the red blood cell is about 10 micrometers, but human cells can go beyond 10 micrometers until 200 micrometers. So that's why all the cells is a good habitat for all these kind of pathogens, including viruses and the bacteria, and so on. All right. So you may see in our human body, we have about more than 10 to the power of 13 of the human cells. And our microbiome, you may see the number of microbial cells, as I mentioned, we call it microbiome, that help us for a lot of digester, immune system, and whatever, as a balance between our body, because we need them for certain kind to get some different like for them to helping us to make up the better immune system or the vitamins. So we cannot say we need to clear all the microbiome in the human. If let's say we clear them off, we need to clear the pathogens, but we cannot kill the microbiome, otherwise the human will die. All right, so you may see the number of the microbiome and the human cells are almost comparable. All right, so even in the fecal, in the stools from us, you may see they have equal number of all this number as well, fecal material. All right, so this fecal, because most of the microbiome can be found mostly in the intestinal. That's why when you pass on the fecal, they come up together with all the fecals. Not all means that this per gram, you know, not all per gram. All right. So this is the, like just now, I would like to give you all some introduction, because when come into the disinfectant and sanitization, our main target will be on the viruses and the bacteria as well. Of course, in terms of living, viruses, no matter how, it doesn't belong to the living organism compared to bacteria. All right, I like reasons I share with you, because it cannot make themselves all the things they need the host to provide the machinery mechanism. For the treatments, of course, we have some antiviral drugs, or we need mostly like the vaccine to try to prevent the infection from the virus. If let's say there's an infection, of course, we need the antiviral drug. For example, the flu drug that we take on is considered as an antiviral drug, and antibiotics. And be aware, antibiotics is used to kill the bacteria. It is not working against the virus. So sometimes we might get confused saying that, oh, if let's say the virus then we take antibiotics, of course it can't work. However, it will make the things become worse, because if let's say we don't have the bacterial infections, but we take in the antibiotics, so in the future, if let's say there is some leftover of the bacteria within the body, so the resistance might be there. So that's why when today a lot of hospital or medical doctors, they are not encouraged to subscribe the antibiotics to their patient. This is a reason why, to avoid the antibiotic resistance of their the organisms. And of course, for the virus, it's super tiny, like I explained to you all before. The size itself is at the nanometer, and this is larger at the micrometer. And for the reproduction, of course, they need to invade or to invest the host to get the machinery for helping them to make the DNA and other like proteins as a capsid for them. However, for bacteria, they can have the binary fuse or split into two, 1 to 2, 2 to 4, something like that. So they don't need any host to help them. They can do it themselves. And the examples, of course, like influenza, like flu, like COVID-19, the SARS-CoV-2, or measles, the polio AIDS. And this is like pneumonia, TB, tetanus, food poisoning, like shigellosis, and so on. All right. So this is maybe I share with you is the difference between these two different organisms. This is how the viruses. Of course, like just I show you. If let's say you have a virus, then they need the host. They start to inject their genetic material into the host, and they take over the host. Then the host will the bacterial genome will help them to make a lot of copies of their viruses materials. Then all this genetic material or all this necessary thing will be formed up together within the bacterial cells. You may see that they have the genetic material formed within their capsid, and they have some protein formed to. This is like the part that they need. So after all this part is formed, then you start to like Lego. You know what Lego they did is they start to combine to each other. They form up each other. Then once they get into the actual form like this, then this part we call assembly and release, or lysis. They will start to kill the cells and release all these baby virus and make more viruses to infect more different cells. So this is the dangerous of how the viruses they infect. So one virus can lead to millions of viruses within the body. Depends on viral load. So depending, but difference from the virus, this is bacteria. Bacteria, of course, it has a genetic material within themselves, but what the genetic bacteria, they can have their own, we call it replication. The name is replication, which means double of themselves. They copy from each other, then they start to form a, we call it split themselves into 1 into 2. So now you have the parent cell over here. Eventually, you have 2 different daughter cells over here. All right. So this is a difference from the viruses. Okay. Now I would like to show you how do the pathogens get into our bodies. All right. There are a couple of different ways. Of course, through the direct contact. This is something that I more refer to this kind of viruses, the direct contact. This is very important when we have the body fluids from someone, infected one, then maybe we transfer to our own body by like this, touching their own eyes or touching their own lips, or all this kind of the route of entry to the body, or any cuts on the hand as well. So it might introduce the pathogen into the body. For example, like touching also, kissing maybe also, if let's say, or animal bites like rabies. You know, any the rabies from the wild dog is less infected wild dogs. Once they bite on the victims, so all these viruses are going through to the circulating system of the body and going to the whole body until to the brain. All right. Another part is a droplet, like for example, sneezing or kind of the coughing. So now the KKM, they would like us to, when you want to sneeze or whatever, you have to use a shoulder to cover your mouth. It's better take a mask, cover yourself, and don't simply sneeze into the environment. Because when the circulation is flying out, you may see like this guy, it can form a lot of different sizes of droplets. For example, when we call it as an aerosol, which means the droplet itself, the size itself is smaller, much smaller than 5 micron or 5 micrometer. However, the aerosol can fly much further compared to the droplets, because it can go further beyond that 6 feet. Compared to the droplet, is this considered, the size itself is more than 5 micrometer droplets. It is less spread, but it can, all this small droplet, because the size itself is much bigger, right? It can fall onto the surfaces and turn out to be one of the resources of contamination, because evaporation rate is much slower compared to the aerosol. So this is the reason why when the aerosol and droplets, sometimes let's say you leave it onto the surfaces like the formats, like our mobile phones or whatever, it turned out to be one of the environmental causes of the disease spread. All right. So as I mentioned, it can be made up from like sneezing, coughing, spit, drool, or even the vomit as well. So this one needs to be aware. So no matter how, putting on mask is still the best way to prevent the disease from spreading to each other. So this is to introduce the difference between the droplets. The droplets, sometimes all this if let's say a droplet itself has been contaminated with the viruses, these viruses actually they will form themselves within the droplets. They're covered by this water, because this droplet to them is kind of like, it's a protector layer for them, because it has all the, not nutrition, but it's something like a protection, because they can fly through, because the humid, right, they need some lower humid for them to fly together. However, when a small infection droplet is, of course, they're still protected, but infection droplet nuclei, certain, for example, measles, of course, until today, nobody found that COVID can be spread through this kind of nuclei, except for the measles, which means that within the droplet, they have a internal layer, we call it a nuclei, which is very, very small, but the viruses can store within this nuclei of the viruses. So let's see, and this, the droplet precursor, for example, if let's say a single infected person, he start to introduce their sneezing or whatever into environment, you may see, if let's say a person stand within a meter with them, of course, he will get infected. However, if let's say the droplet itself is belongs to the aerosol or within the confined or non-event, we call it as within the aircon space environment. So all this will go through to the system and introduce to others. So need to be aware. So usually we refer to saying that a lot of the crowd activity need to be avoided. If let's say there's a need within the crowd, of course need to be having a social distance, and try to do it not within in the confined space, or the air need to be ventilated more within the area. All right. So the droplets, all this kind belongs to the surfaces that might be exposed to the droplets. For example, the contaminated droplets might be exposed to the surface of the, mostly on the upper surface, because this is what our living organisms, our human will use, right? So this is surfaces, or the table, chair, or the toilet itself is quite serious sometimes, because usually a lot of things that all our passing activities is done in the toilet. So this one need to be aware. And of course, on the handle for this kind of like the escalator one, even in the lift buttons, or the door knobs or whatever. So this considered as all of the sources of the contaminations. So when we try to do the disinfectants, we have to be aware of this kind of the surfaces mostly, rather than in the air, because as I say, COVID-19 or SARS-CoV-2, it can be only introduced by an infected through the air, but the virus, there's no evidence saying that it can be stay in the air like measles for long, which means that usually they will be killed. Maybe if let's say it's like in Malaysia, it's very hot, right? They will kill after drying up very soon, maybe in a couple of minutes. But we still need to be aware, because we don't know who is passing by around us, that they might be the vector for the disease. All right, so this is, I would like to introduce is airborne transmission. So because COVID-19 doesn't belong to the group of airborne transmission yet at the moment, because no evidence found it yet. However, we know that it can be introduced through the droplet. Airborne transmission germs, which means that they can travel across the confined room or through the ventilation system. All right. For example, what are the airborne transmission organisms, like measles, or morbillivirus virus, chickenpox as well. For example, if let's say using usually, how come sometimes we get the chickenpox within the class, right? Because someone getting the chickenpox, so the virus can be transmitted through the airborne within the air, within the confined space, and even the TB, flu as well, and enterovirus like hand, foot, mouth disease within the usually happen to the school the small child. All right, so ventilation system need to be aware. No matter how, so when we do the cleaning, of course, you cannot, no matter how you clean the whole organism, we cannot see the virus, right? So the most important we still need to clean is about the surfaces, and keep the hygiene, which means that put the mask on from time to time, even within the confined space, if let's say the crowd is there. And most importantly is we need to keep the social distancing, all right? And if let's say, maybe I'm not going to touch that on the HEPA filter one for the aircon, we need to keep upgrading the HEPA filter or change the HEPA filter from time to time to avoid, to make sure that it can filter the air, because the air will be filtered through HEPA system before it bring out to the aircon, right? In the ventilation system. So HEPA filter is very important. Okay, fecal oral transmission. For example, fecal oral transmission usually is, of course, some study found that the SARS-CoV-2 virus or COVID-19, I call it COVID-19 virus is better because people know that SARS-CoV-2 is the name of the virus. Let's say people say that SARS-CoV-2, COVID-19 virus can be found in the, of course, from our droplets, and certain study found that it can found within the stools or the. However, there's no research saying that the stools belongs to the agents of transmission yet at the moment. So don't worry about that. However, because this virus, it can bind to a lot of receptors. We call it ACE2 receptor within our cells in the body. So because ACE2 found along the way down to the our lung esophagus, this esophagus connecting to the lung, it also can be found in our heart, and can be also found in the intestinal ACE2. It's one of the protein receptor. So that's why when there's a particular receptor, then these viruses will attach to that and start to do their infections. All right. So no matter the fecal oral transmission, we need to be avoid also, not only for the COVID-19, but also for other particular diseases. For example, the germs can be introduced through the someone maybe they never clean their not touching the stools, but they never clean their hand properly. They're touching somebody else, the contaminated stuff, right? So they introduce you by eating something in their mouth, all right? So they cause vomiting and diarrhea, and commonly is infected by somebody, they don't wash their hand properly before using a bathroom, or they never wash their hand properly when preparing food to others, all right? So this is maybe today, I think most of us, they are quite interested to know about what happened if, let's say, instead of having from person to person interaction, and is there any things like vector can cause to the bacteria or viruses infection? Of course, yes. The answer is yes. The insect considered as one of the major agent or one of the major vectors to transmit the bacteria or viruses to the human as well. For example, this include like a West Nile virus, or WN virus, I will call it WN virus usually. Dengue virus, malaria. We know, right? Malaria is through the mosquito as well, and chikungunya viruses. All this can be introduced through the different types of the mosquitoes. Of course, through the biting, because all these viruses, they store themselves within their salivary gland over here. So when the injection is done through within the human skin, so the viruses will pass through from the injecting tube into the circulating system of the human. Okay, this is the recently news that I think most of us been going through, right? Recent news that I think 2 days ago, yesterday or today, what's the date? 5th of August, I think, 2 days ago, found in one part of the China as well. You may see it's a tick bites. One of the viruses, we call it as SFTS virus, severe fever with trouble, is quite long. This name, we call it a Bunya virus, or in the short form is a SFTS virus. It can be introduced through the bite of the ticks. All right. So this kind of the ticks is usually found maybe not in Malaysia luckily, but we don't know because it's a small size. So this is a female and a male. So it is potentially to introduce these kind of viruses through the biting into the human. So this is quite serious case also. So we need to be aware. And these viruses, usually the impact is not like COVID-19. However, it will form, you see that the thrombocytopenia, which means that you form people will get inflammation in our blood system, blood circulatory system, and it leads to the death as well. So that's why there's 7 of them reported death after identified with this kind of the virus infection. So this is I'm sorry, because the figure itself is quite small. You may see along the way, since the discovery of, I mentioned, discovery of the bacteria or the pathogens, until today, and 2020, but this until to 2010, it's fine, it's roughly the same. You may see there are a lot of the pathogens has been found over the years, and a lot of vaccines also has been developed to try to control all these organisms. Unfortunately, for example, certain things like you may see some asterisks like influenza. We still developing a kind of like effective. Although we have the vaccines, however, the vaccine itself is not. We need to have a kind of like update vaccine from year to year. Just because of influenza itself is quite complicated. It has a eight strands of the 8 segments of the RNA within their within their genome. That's why all these DNA, they can do the recombination from year to year. That's why the vaccine need to be adjusted from year to year to try to make it comparable to the latest version of the flu. Also, so which means that these are pathogens, but luckily we have the vaccines and as well as the suitable drugs. We can control a lot of like diphtheria, TB, or pertussis, hepatitis. We still can control them easily. However, the current one, like COVID-19, because it belongs to a very novel disease, of course, times is needed for a vaccine, an effective vaccine development. All right. So now let's look at this coronavirus. Actually, it's not new not new. It's not a new term today. The first SARS is quite important, that found in about 10 years ago in Hong Kong. It was because SARS-CoV-2. This was also sometimes because SARS-CoV number 1. It found in 2002. However, the mortality rate is much higher compared to our today's SARS-CoV-2. You may see it's about the 9.6% out of the 8,000 total cases. But it was lucky no cases was reported until 2004. However, in 2015, another coronavirus infecting case again happened in the Middle East, which means the MERS stands for Middle East Respiratory Syndrome. This kind is a respiratory syndrome, coronavirus. So which means the mortality is much higher, is up to 35%. However, the infecting rate is lower compared to the SARS. So also through the limited human to human transmission, but this is usually it was found is through the camel in the Middle East. Then they start to pass to the human, and human to start to pass on to others. And today is SARS-CoV-2, but luckily, of course, the transmission rate is much higher compared to the other type of the SARS-CoV and MERS-CoV. However, the casualty rate is luckily is not that high, which means that it's about less than 2%. As compared to this is up to 99.6%, this is up to 35%, but this is less than 2%. But we still need to be aware, because the severity is still there. All right, because it has the potential to pass on to one to another, and we still need to be aware of this disease is quite risky also. So when we look at this diagram, it come up with an assumption to showing that you may see that for about every single 10 years, the coronavirus turn out to be a new species again. So which means that if let's say we don't control the coronavirus today in 2020 or whatever, in 2030 again, roughly, we may come up with another type of coronavirus through the mutation or whatever, because the coronavirus, they are not found originally from the human, but they are found originally from a lot kind of the organism. It can be from the, like we know, today we say from the bats, right? It can be from camels, from a lot of things, from even from some of the study we did before is it can be from the rodent, from mouse, from the chicken as well, a lot of different organisms. But the thing is, typically, once they settle themselves within the host, usually they won't spread to others. However, if let's say mutate, because virus itself is very simple form in their genomes, so if let's say a single mutation happen on their genome, so it can make them to be transmitted to the human through the genome recombination. All right. So this is a transmissibility and the fatality rate of the SARS-CoV-2. You may see that SARS-CoV, for the first one, the SARS found in Hong Kong years ago, the R0 is quite high, which means the transmission rate is very high, 2 to 5. And MERS is much lower, less than 1. For SARS-CoV today, the transmission rate is pretty much similar to the SARS-CoV found in Hong Kong, but luckily the casualty rate is not that high compared to the first two. But nobody knows. That's why we need to control, because we may see the case like this, right? It can happen like every single 10 years. We do not know what's going on. If let's say it has any mutation, but luckily not until today. Luckily not. All right. So what are the environmental factors that would affect to the transmission of the SARS-CoV-2? So we may found that some study found that, of course, the temperature. The higher the temperature or the higher the humidity, like in Malaysia, you can see that our transmission rate in our Asia country is, or in Malaysia, Southeast Asia, is not that bad compared to, except Singapore. Singapore is because of the, I think because of the overcrowd already, the population is too much. So you may see the transmission rate is still much controllable compared to Melbourne. You may see like recently the case in Victoria or the Melbourne is start to increase again, just because of now Australia get into their winter season, especially to Victoria, because towards to the south. So the temperature is very low, and humidity there is comparable low like now. So that's why it is potential, because of the disease, the virus that can spread much better in this kind of the environment. Of course, the air transmission, like just now, like the droplet, everything it could be one of the environmental factor. However, insects, initially people just suggested it, but there's no any evidence saying that insect might be a vector for the coronavirus to spread to each other. But it can be the resources. The insect can be an agent for a vector to transmit other viruses to the human hood as well. And of course, the surfaces, all these surfaces, like all the surfaces I explained just now, it can be a major source to transfer to transmit all the disease to the human. All right. So let's look at some structural differences between the viruses and the bacteria. Of course, this is this I show you is the COVID-19 bacteria, COVID-19 virus. Of course, you may see there's a genetic material within that, and it is stored within their core, or within the nuclear capsid over here. And outer layer, they have this we call it as an envelope, which means that it has something like the cover, the lipid cover outer there. You may see all this belongs to the lipid layer or glycoprotein over here. Okay, compared to the bacteria's shape is pretty much similar, but it's a bit complicated, which means it has a genetic material or genome within themselves. You also have a external DNA bacteria, we call it as a plasmid over here. And depending on the I just say the common bacteria, it has a layer out there, we call it a plasma membrane, and it's covered by another membrane, sometimes we call it a cell wall also. But this flagella and PD is depend on one bacteria to the other. Some of them they have it and some of them they don't have it. All right, but this is what I want to mention. This is so the difference is, of course, it has complicated genome. However, this is have a very simple DNA compared to the virus. And then both of them also have their own outer layer membrane to cover them. This we call it as like a envelope proteins or capsid over here. And this one we call as a membrane, a plasma membrane, and covered by a layer of the cell wall. All right. So when look into in detail, this is a difference with bacteria. Bacteria, sometimes because we have two types, or we call it gram positive and gram negative. Of course, I'm not going to touch too much about how are we going to tell the difference between these two, but I just want to share how are we going to introduce, because when you look at the disinfectant, they will say it works against the gram positive, it works against the gram negative, but we need to know what's gram. The gram itself here is from the name, it's not from the kilogram, the weight that we mean. Gram positive, which means they have a thicker layer of the peptidoglycan over here. All right, this thicker layer. So, and then this is the internal layer of the bacteria, which means that the whole bacteria itself is covered by a layer of peptidoglycan on the outer layer, followed by a plasma membrane. All right, from gram negative bacteria, which means they cannot stain through using the gram, that's why we call it gram negative. It doesn't mean that nothing. Negative, which means we cannot stain it using the staining method of the gram over here, so we call it gram negative. However, this one, it has a peptidoglycan over here as well, but it's still covered by a layer we call it outer layer, outer membrane. All right, so this is the difference between the. So usually in terms of strength, gram positive bacteria sometimes is very hardly to be killed because their peptidoglycan is much thicker compared to this gram negative bacteria. All right. Okay, for the viruses, of course, when you look into the disinfectant, sometimes you will mention that it works against the envelope virus. And what is non-envelope? So envelope virus, which means like the coronavirus, they have a layer of the outer layer over here, lipid layer. All right, this covered the capsid, and then the DNA is sitting within the nucleocapsid over here. The non-envelope virus, which means it doesn't have the envelope out there, but it has a thicker outer layer as a capsid for the protection. So in terms of when you see that these envelope virus, actually they are more weaker, or I can say much vulnerable compared to the non-envelope one, because of they are sensitive to heat. And then compared to the non-envelope, they are quite resistant to heat because it has a thicker capsid formed by this. All right. So why do we need to use the differentiated disinfectant? In this case, I try to use the common one is alcohol, because usually we use alcohol to try to disrupt the membrane of no matter it's the viruses or the bacteria, because bacteria and the viruses, let me show you. They form by a layer, we call it as here. You have this is out here is a lipid layer, and the tail here is a protein layer. All right. No, sorry. This is a protein. Within the inner side is a lipid layer. That's why we call it a phospholipid. Phospholipid layer because of this kind of the combination. For the alcohol, usually it's recommended to use a water diluted alcohol, just because of we need to form two kind of substances here. One is a hydrophilic water living head, which means to target against the protein head, and another one is hydrophobic or water hating tail. This one will be targeted by the alcohol. If let's say certain people they say if you use a higher percentage of alcohol, it can work, because higher percentage of alcohol, it doesn't have much hydrophilic part, so which means that they cannot penetrate through the outer layer over here. They cannot through the outer layer. So once they cannot penetrate to the outer layer of the membrane, so the alcohol, no matter how strong it is, they cannot get interact with the hydrophobic part of the membrane. So which means it can't work at all. So that's the reason why we need to use a combination like a 70 to up to 80% of the alcohol to ensure that they can interact. The water can interact with the protein to the head part to tear off the head part and let the alcohol to penetrate into the internal layer, so it can cause the cells disrupt. Once the membrane is disrupt, and consider the whole thing is gone already, so they cannot survive. So these are the common things that we share together from a lot of today's health authorities. Of course, all these pathogenic bacteria or virus, they can be killed using the hand sanitizers or hand hygienes. Clean it properly, so we can inactivate the pathogenic lipid membrane and let them to release their intracellular contents, so they cannot survive anymore. So this also happened to bacteria and to viruses. All right. Okay, I will stop for not right now. Maybe I continue this first, then we stop in the next slide, because this is just 2 slides only. So before we would like to, so I'm going to share with you the difference between the sanitizers and the disinfectants that we know today. So the grouping of the chemical disinfectants. Of course, we need to understand 3 different types. There are 3 different, we call it as germicides. Germicide, which kill the germ. Site something is something like killer. We kill the germs one. The high level of germicides, which means some group of the sanitizer or the chemicals can be used to kill most of the living organisms, including the vegetative cells or vegetative bacteria or fungi and viruses. And this most important is endospores. All right. So which means that usually when talk about the high level germicides, it came up something. The idea is like a sterilization. It kill everything. In the second level, we call it as like intermediate level germicides, which means they cannot kill all the viruses, but selectively, maybe they can kill better against the envelope viruses, but not for the non-envelope viruses, and they are less effective against the endospores. And for the low level germicides, which means they can kill most of the bacteria as well, and some selected envelope viruses, but are ineffective against the endospore. All right, but today, what we need to use in the market here, we still need to focus on the low level here, because luckily, maybe I want to show you this diagram. You may see this is the pyramid, right? So this is more susceptible, which means easy to kill. This is hardly to kill. You may see that the coronavirus, luckily they all sit at the bottom of this pyramid, which means that they're easily being killed. Even they use a heat, you use a normal sanitizers like alcohol, whatever, or any kind of the disinfectant, suitable disinfectant, we can kill them easily. All right. So compared to certain kind of like bacteria, for example, you may see the certain fungi, because they have a thicker layer of them, so it is hardly to be killed. So we need to use that intermediate level. And even certain bacteria like Clostridium, they can form the spores one. This one they cause the tetanus. So this is hardly to be killed. Even like TB also seated in this area, because they can form something on the outer layer is much thicker, hardly to be killed. So this one usually we have to kill them using the heat or using the strong chemicals like formaldehyde or whatever. But today, because luckily coronavirus, they are seated underneath the pyramid, so they can be killed using the normal. I can't say normal. We can use the low level or germicide, so to kill this kind of envelope virus. Okay, before we want to understand or want to proceed to the next chapter on the chemical differences, we need to understand what's a sanitizer. Of course, we keep on saying that we know that sanitizer, hand sanitizer, but the terminology sometimes we get confused. So sanitization, which means that a certain process to reduce the microbial contamination to an acceptable level. All right. So the process of learning, cleaning objects without necessary surgery to going through the sterilization. This is something as long as we reduce the microbial contamination. So sanitize is not as strong as the word of disinfectant. Disinfection is a word that is a procedure of treatment that eliminates many of the pathogenic organisms. All right, with the exception of certain bacterial spores, which means that the spore itself they cannot kill, but you will kill majority of the bacteria in the environment. The safest way is still the most effective way to kill all the organisms is still using the sterilization, but this is harder to be done in the environment, because usually at sterilization we need to have a setting. Like for example, you need to use a heat, or you have to use a very harsh chemicals to do that. But harsh chemical is quite dangerous. Is quite risky to the person who handle it or to the environment, because it can a lot of residues may left on the after use. So this is considered as complete elimination, 100%, to kill all forms of the living organisms. All right. So because my slide is up to the, maybe we still have roughly 50% to finish. Maybe we can have a 50% break over here. Not 50%, 5 minutes break over here. Then we can continue again. Any question, you're welcome to share with us here. So we try to share with you, I'll try to answer it at this slot within the 5 minutes. Thank you very much.
Okay, thank you, Dr. Leow. Okay, everyone. I don't see any questions being raised at the moment. I think because it's more theoretical right now for the science and all that. So they may have more questions later. So I think let's just take the 5-minute break right now. It's now 11:05 on my clock. So please come back in 5 minutes time at 11:10. Yeah, so thank you, everyone. Just take 5 minutes, take a break, and then we can come back again to continue the session. Okay.
Thank you, Chris.
All right. Okay, thank you everyone. Hope you managed to have a good break. Okay, so it's now 12 past 11. We still have quite a bit to cover, so I'll just pass the time to Dr. Leow again to resume his topic. All right, Dr. Leow, over to you.
All right, thank you very much, Chris. Chris, would you mind to let me know whether you can see my slide over here again? Because
I just stopped the share from my end, so you just need to click share again on your side.
Okay. Oh, I have to. Okay, hang on. Thank you. Okay, so it works, right? Hello, Chris. Does it work?
Yep, it's okay. Sorry.
Okay, thank you.
Yeah.
All right, because I tried to, now we finish up just now was the kind of like introduction. We need to know some terminology, of course. But now we get into the second part, which means that I'm going to share with you about what kind of the disinfectant or the chemicals that we use in our current disinfection procedures. For example, the alcohol, of course, alcohol considered as one of the major chemicals that we use mostly in today's disinfection. Of course, this one can be formed by ethanol or the isopropanol. Or that it works against most of the living organisms, including the bacterial, fungus, and virus living or enveloped viruses. But of course, it's not working against the spores. But luckily, because today we are not working against this kind of like Clostridium, all this kind of the deadly bacteria forming the spores. So it's against the viruses. So alcohol itself is considered as a most effective way. So as I mentioned just now, the best alcohol concentration to be used against to kill the living organisms is within the 70% until to 80% and diluted in the water. All right. So another part is, of course, chlorine itself. Chlorine is considered as a very strong oxidant because it provides the broad spectrum as to kill most of the living organisms. So the working concentration for the chlorine, we can get it easily from the market, is from the 0.1% and to 0.5%, depending on the situation. Usually, if let's say within the indoor the confined space, usually it's recommended to use a 0.1%. However, because the chlorine itself is very highly alkaline and it leads to the corrosive to the metal, and it could potentially to decolorize the fabrics. So it's recommended to be stored the chlorine at the room temperature to avoid the releasing of the chlorine gas, because the chlorine gas itself is very highly toxic, and it can lead to our problem in the breathing system, and even to the death as well, chlorine. So sometimes chlorine is not highly recommended to be used within the indoor unless you have a good ventilation system. So three products that never mix with the chlorines. For example, when we prepare, so need to be aware, we need to get the instruction to be studied exactly, or follow exactly MSDS. Never add any kind of different ingredient into your chemicals for the disinfectant to be used. For example, don't add the ammonia, because after ammonia is added to the chlorine, it forms the chloramine gas. This one will cause the coughing or shortness of breath, or even the pneumonia, because shrinking in the lung. And the vinegar as well, acidic compounds. So never added to the chlorine gas, because this will form the chlorine gas. Never add with this kind of bleach, or they form the chlorine gas because of the gas, right? So this one also will cause the chest pain, vomiting, or even the death. And finally, never ever add any alcohol into the bleach, because it forms the chloroform. Chloroform, this will lead to the fatigue, dizziness, even fainting, and even death as well. And this chloroform, most importantly, is once chloroform is formed, this one considered as smellless. You feel nothing at all. So this one need to be aware. All right. Okay. Another thing is another strong oxidant that we use today is a hydrogen peroxide. Usually the working concentration we need to prepare is a 0.5% to 1%, but in the market usually is up to 3%. So depending on what kind of so when we want to use it, either you can use it ready to be used, or we can dilute it down to 0.5 to 1%. But because itself is a good decontamination agent, however, we need to be aware compared to chlorine is still corrosive, but not that much corrosive to chlorine, but still considered corrosive, depending on what corrosive against the metals such as aluminum, copper, and zinc as well. And also will lead to the decolorization of the fabrics, maybe irritation on our skin if let's say high concentration is used. And so need to be stored properly to stay away from the heat, and need to be protected away from the light to keep the efficacy of the hydrogen peroxide. Because when the light is there or the heat is there, then they will break down the H2O2 itself into the hydrogen and oxygen. However, the toxicity is not that bad compared to the chlorine. All right. So another agents that we use mostly today also like Dettol, or sometimes we call it a phenolic compounds or chlorocyanero. Of course, it belongs to the earliest germicides. It is used to kill most of the bacteria and lipid viruses and even mycobacteria. And of course, itself is corrosive to the skin. So sometimes it's don't use it to higher concentration. And if let's say the chlorine itself, the phenol itself is not well prepared, it will form into the benzene. Benzene, this is quite dangerous because it leads to the carcinogens. So it's not recommended to use as food contact surfaces, and try to stay away from the young children as well. That's why phenol or this thing, we try to not to be used in a huge environment. So we still prefer to use something like conventional disinfectant as well. Another part is sometimes we know it as quartz. Quartz here usually we call it as like the chemicals compound is benzochromium chloride. So usually it's more than 200 ppm. Then we use it, if let's say used together with the alcohol solutions, so it shows the efficacy to kill the non-enveloped viruses like the COVID-19. So of course, this quartz has been widely used in the industrial water system to try to reduce the organic materials there. Okay, last but not least, also some chemicals that we use, I think most of the professionals, so here we know is a formaldehyde. So formaldehyde is used to kill most of the organisms and the spores, and it works perfectly. Even the 5% formalin in water are used as a liquid disinfectant. But one thing is formaldehyde itself is very, very toxic, and this is harsh chemicals. So it need to be used in a well-ventilated system, and itself is a carcinogenic. So usually we need to get the license to handle with this formaldehyde, and a place has been disinfectant with the formaldehyde need to be noticed, because some residues is there. We need to check. We have to check how much residue left over before it can be used by the public. So formaldehyde, to be honest, is not highly recommended to be used like in the fumigation, because it used to kill the insect, but not used to kill for this coronavirus. So this account that I get it from one of the CDC. You may see the alcohol, chlorine, this iodine-based disinfectant, phenols as well, and the quartz. You may see that effectiveness is quite. They are very effective against envelovirus like coronavirus, even some gram-positive and gram-negative bacteria. But because when you come to the later, like the spores itself, this is quite, I said right, it's quite harsh to be killed. So of course we need to use a much better or much stronger disinfectant. But today I will focus on this kind of the viruses. I think all the chemicals listed are suitable. But one thing before we purchase, of course, we need to get it from reliable company, that it can tell us most of the MSDS and the instruction. All right. Okay. This come to my last slide, which means that we could not last slide, last part of the slide, which means that we're going to show you what kind of the environment that can be having the potential exposure to the microbial contamination. For example, the surfaces in the office, public area, the chairs itself, in the public toilets, or in the shopping mall, all these handlers, even the steel bar, even in the kindergarten, in the schools, or a place that has been touched by a lot of different touched by the human together. So all this considered as a potential resources of contamination for the pathogens as well. So there are some studies to show that the wood and the cloth, the sustainability rate for the COVID-19 virus to live on. They can be living on this, if let's say not like this skin, a contaminated chairs or the wood, they can be live on for up to 1 day. And they can even live on the surfaces of the glass or the aluminum bar there up to 2 days for the virus. And for the stainless steel or the plastic, they can live on for up to 4 days. But these are all the experiments done not within the clinical setting, even the environmental setting, but they did it in the lab setting only. So this is just for references. So as long as you keep ourselves clean, so don't worry anything else. So we just most of them, we have to keep all the surfaces, must keep all the surfaces clean from time to time. Okay, how the first thing when we start to say about the cleaning, I want to show this slide again because we know that the bacteria will form a layer we call it a biofilm. Of course, this biofilm can be found in let's say our teeth is not cleaned properly. This one we call it biofilm. The biofilm actually is a secretion from the bacteria for all the different kind of bacteria to live on together. So once the biofilm is formed like this, so the bacteria itself they will get protected from the environment. So once the redundancy of the bacteria is reached, then they start to release and go to another new area again. Maybe one of the area I can show you is like in the base of the bathroom. Sometimes it's not well cleaned. You may see some certain like pink color is formed over there. Actually, this is the formation of the biofilm caused by the bacteria. And underneath the shampoo bottle, every time you can see something, if not clean, they may see some pink color there also, near to the water tap and within the sink. So this all belongs to the biofilm formation. So if let's say you want to clean a place properly, all this biofilm must be cleaned off first before you want to apply the disinfectant against them. So how are we going to do that is, the first thing, we have to do the cleaning, we need to use a proper water. Of course, water is needed. Use a soap or the natural detergent. Then mechanical action like brushing and scrubbing. This all will not kill the bacteria, but you will try to take off their territory. So to let them exposed, when you use a disinfectant, they can be applied straight away to all this clean area to kill all the bacteria there. So because we would like to use the way to try to remove the organic materials. Because all this kind of all these biofilm they form because of the organic material left over on the food. That's why they can survive. So because we need to remove all the organic material to inactivate the the organic material can inactivate the luminescent side of the properties of the disinfectant. So we have to remove them completely first before we use our disinfectant. And when we come into the disinfectant, of course, the concentration and the contact time is very important. I think this one we can know that from the instruction of the manual when you purchase. Usually concentration, say for example, for the hydrogen peroxide, we recommended to use like about less than 1%, 0.5% to 1%. And contact time all are same, must be within at least a minute, even for the alcohol, within a minute. So the longer it exposed or interact with the organism, so the more they will kill. So we have to apply after the cleaning to kill the remaining microorganisms. And but be aware, because the concentration is play a major role over here. If let's say insufficient of the dilution, it might be the effectiveness might be reduced. However, if higher concentration is used, it might damage the surfaces of the furnitures. So this one need to be aware when we use all these kind of disinfectants. So last but not least is to show you when we try to practice these disinfectants, the first thing is, before we want to try to help others or we try to save the environment from to be safe, we have to make sure ourselves is safe from when we're doing our cleaning. PPE must be practiced from time to time. So what does it mean with the PPE is, of course, this one has been, all the while we know that KK authority, we try to highlight it. We need to have proper goggles, all right, or the face shield together. You need to have a 3-ply face mask together, and a long sleeve gown, and rubber gloves as well, and a plastic apron, and rubber boots. That's all. And then after use, they all need to be put off using a safer way also, not by touching your own hand, the naked skin, by using all this contaminated stuff. All right. Okay, thank you. That's all for my today's slide. I hope that you all may learn something new, differently within your area. So if you have any question, I'm happy to answer you from time to time. Thank you very much. Thanks, Chris.
Okay, thank you, Dr. Leow.
No worries.
Okay, hang on. I'm just going to share the screen again to enter Q&A. Okay, everyone. We hope that you had a good session. So now it's time for questions. Let me read out the questions for everyone, and then we get Dr. Leow to answer them live. So I have one question here from Michelle. And she's asking, wait, hang on. And she's asking, is it recommended to use 60% base alcohol sanitizers or soap with water for COVID-19? Yeah, I don't know. So is it safe to use non-alcohol based sanitizer? Yeah.
Yeah. Of course, it's safe. I think the 60% itself is, to me, I found that 60% is not highly recommended at this moment. Maybe you can use slightly higher, like up to like 70% is the best way, because even in our laboratory setting and in our clinical settings over here also, we use like at least 70% and up to like 80%. But most of the time we use 70% or 75%. 60% usually sometimes we worry the way of preparation, because maybe if let's say too much of water is added again, it may be reduced down to even lower than 60%. All right. So this is a first question. The other question is it safe to use?
The second part of the question is it safe to use non-alcohol based sanitizers, and does it carry the same safety?
Thank you, Chris. I think this is a good question. I will say that we can use the non-alcohol based sanitizer as well. For example, there are a lot of the different sanitizers now prepared by using non-alcohol based sanitizer, for example, made of like H2O2, the food grade hydrogen peroxide, something like that. But of course, they are more like using the quad as well. So which means they are not alcohol based. So they also carry the same safety like what required by the health authority as well. Don't worry, it still can kill the normal envelope viruses.
Okay, all right. Thank you. So I have one question here that's asking about sterilization. So with regards to sterilization, does it mean that when you do sterilization, it must come with additional treatment like heat instead of chemicals? So we need to clarify a bit more about how sterilization is classified.
All right, cool. Thank you, Chris. I think, sterilization, usually they can be divided into two parts. One is a physical treatment, the other one is a chemical treatment. Physical treatment, something most of the time we will use the heat to kill them. All right, but all these heat setting usually hardly to be found in a normal sort of whatever. Usually we can get it either in our laboratory or in a hospital, because all the surgical set, right? When we wanted to use apply onto the patient, they need to be clean to make sure the sterility is reached. So the heat one usually is quite limited to the certain area. But for the chemical wise, the chemical sterility is just like I mentioned in the slide, is usually people will use like formaldehyde to kill all the germs over there. But there's a pro and cons using the chemicals. Because the chemical itself is so toxic. So if, let's say they are not well prepared, it will cause a problem to the one who prepared it, or the residue left over onto the things. If not, say they're not cleaned properly, that will create a problem to the next person who use it. So sterilization, usually people still prefer to use the heat sterilization. It's much safer. Yeah.
Okay, thank you. Yeah. Okay, I have one question from Mr. Kanis. He's asking, some people's skin are quite sensitive to, for example, to ethanol, for example. So is there any possibility to use other added solution with ethanol to produce sanitizers?
All right, this is, thank you very much. This is a very good question, because certain people, I know that alcohol may lead to the allergic to someone. So sometimes we know that because in the market today, we have quite a number of different kinds of sanitizers we are using. Alcohol, actually ethanol itself belongs to one type of them. If let's say ethanol itself is not suitable, they can try to opt for other options. For example, like just now I mentioned, like hydrogen peroxide or something like that, but it need to be food grade. All right, food grade, which means that it's suitable to be used by the human. And but we use according to the instruction, maybe like 0.5% should be enough. and then they also can try to use that the quartz material like benzochromium. Benzochromium also another part of the things, like maybe they don't need any alcohol to be mixed. It can be mixed or without. So, but without alcohol, it's still working. So they can try to look for some alternatives for them. Yep.
Okay. So he actually has another part to the question as well, because he's asking also are biocides suitable for this?
Biocide actually is the terminology is quite wide. I don't know how could I to tell this Mr. Carnison about the biocide, right? Because everything that can be used to kill the germs, one we call it a germicide, but biocide is much wider. So I don't know what does it mean with the biocide.
Okay, so Mr. Carnes, if you are hearing this right now, maybe you can clarify what sort of biocide. In the meantime, I'll move on to another question first. So I have Fazila here is asking, based on current figures on COVID-19, so she's asking, how many more you think people will be affected until the vaccine is produced? I think this is quite a big question.
Yeah, but this is a question, I like this question also because we have this question when we have our scientific discussion, right? Because to be honest, a lot of pathogens, the vaccine is not found yet. For example, HIV, SARS coronavirus, even for the SARS-CoV-1, like I mentioned just now, found in Hong Kong years ago, and MERS, no vaccine at all against it. And malaria, we don't have vaccine also. But how come we can control ourselves not getting HIV, not getting malaria, not getting this thing from time to time? The first thing is our own hygiene is very important. For example, now, let's say now it's a pandemic season, right? Everyone, when we are asked to wear the mask properly, not underneath the nose or whatever, we must change the mask and wear it properly. We must follow because this is a pandemic and the disease is around us anytime. So to reduce the chance of getting the disease, so the first thing, if we follow the hygiene, the second thing is if let's say everyone is free from getting the germs, so which means the whole environment is safe. It's similar to the to other diseases like malaria or whatever. We have the dengue. We have the dengue in Malaysia from time to time. Dengue is, we don't have a particular vaccine against them yet. But we have the mosquito. We hardly to control mosquito, right? We may need to ask our expertise, a lot of expertise why she just call her Siu from time to time. So yeah, because Karen. So we ask Karen because we cannot control most of the mosquito first. So which means that even though we don't have vaccine, as long as we keep ourselves good practice, we can still keep ourselves to try to pull down the number of the case to the minimum. Yep.
Okay, thank you. Yeah, I think this this question also is not so easy to answer because none of us can predict what's going to happen next. So like you said, I think we really just have to continue to be careful and put best practice in place to keep everyone safe whilst a vaccine is being developed, right?
Yeah, right. If the vaccine is developed, it doesn't give our 100% security as well because like the flu vaccine, right? Because flu vaccine need to get updated from years to years. So it doesn't mean that this year I get myself with the flu vaccine, next I will be safe myself from flu forever. So we still need to be aware to not be like keep the hygiene properly. Yep.
Yeah. Okay. Okay, I have one question here asking to control the environmental growth of microbial growth, right? Do we have to monitor the pH and the temperature of the environment? For example, the environment in a building,
Oh yeah, good. Thank you, Chris. Yeah, of course. Sometimes we need to. Of course, all this. If let's say we measure pH, might not be necessary because let's say if you want to measure usually pH, we measure the solution, all right, in the solution or in the water, in the swimming pool, whatever. But let's say you mean the air environment, we will try to get some information about the humidity first, the temperature, the humidity, so that you will know that how are you going to use your disinfectant to try to target around the whole environment. Yeah.
Okay. Thank you. Yeah. Okay. Here's one more question about. I think this is about technique. So when it comes to disinfection, right? Is the technique supposed to be wipe, mist, and wipe again, or is it wipe and mist, or is it just misting alone? So which method would be recommended when it comes to disinfection?
You mean what bite do you mean?
So the question here is about how to do disinfection, right? So the method lah, right? So the question is asking, is the correct method wipe, mist, then wipe again, or is it just wipe and mist, or it's just misting? You know, so there
Okay,
are
good.
three techniques here. So the question is, out of these three, which method is recommended?
Okay, to be honest, the proper method recommended by all these like the CDC and WHO, all this, they mentioned that of course we must clean the area first, and we clean, which means that like just now I mentioned, we have to wipe off the area using kind of the soap or the detergent to wipe off the area first, then start to apply the disinfectant against. Because to be honest, if let's say a surfaces itself is covered by a layer of the organic material, no matter how much of the mist or how much of the disinfectant you try to apply on them, it can't work at all. Because this organic material they will reduce the efficacy of the disinfectants. So the method is we must wipe that off first using the clean water with the detergent, wipe that off first to clean off the bacteria, clean off the organic material, followed by using the disinfectant. This disinfectant, if let's say you want to use, we recommended to use the wiping method because it has more surfaces to be to evenly to wipe on the surfaces. If let's say the surface itself is so wide that it's impossible to be using the wiping method, of course we can use a misting method. But when we're using a misting method, you need to be sure that all the mist, when you need to be rolled evenly and slowly to make sure the whole area itself is exposed to the detergent so that it works. Otherwise, it can't work at all. Because sometimes we you know, especially near to the corner part, let's say the misting itself is not sprayed onto the corner part, so we the area itself is still not fully yet decontaminated.
Right.
Okay. Can I interrupt? Actually, that question is from me. So I want to continue some more. Just now you mentioned if let's say the place, the surface area we can wipe, we just use wiping method. If let's say it's a very big one, we use misting method, but we must make sure it's evenly misted at the surface. Then after we mist, do we need to wipe it again? Because it contains chemical. Let's say we use quat. Quat is actually non-alcohol, right? So after we mix it, do we have to wipe it clean with clean water?
Not necessarily, because quat itself, I think, is quite suitable to be used within the human because it doesn't belong to the harsh chemical, right? And some more, like all these H2O2 quat itself, they usually, after you using, then when the time progress, the evaporation's done, so all these chemical automatically, they will be gone, something like that. It doesn't seem like the formaldehyde or formalin. So the molecule will still, the residue will still stick on will still left over, if let's say even dry up. So this is quite dangerous. Yeah. But for this alcohol thing, benzochromium, I don't think so they have any chemical will be left over. Yeah. Okay. Of course you can, the wiping itself is not for to wipe off the chemicals, but the wiping itself is the way to try to make it evenly spread all the surfaces on it. Yeah.
Oh, so okay, so that means we mix and then we leave it dry, and then it is safe for people lah.
Yeah. Okay. Usually what we need to do is we will clean it first, then you do the misting or you wipe it first, then leave it for a while, make sure the ventilation system is properly controlled, then maybe wait for half an hour, then we ask the people to come in or whatever. Yeah. Usually, even like today, I told you, right, we were having a cleaning in a short while here, so I'm not allowed to sitting in my office. So then they will leave it for a while. Then maybe after a couple of hours, then they will open it again for us to come in. Yeah.
I see. Because in our industry, when we talk about misting, we use pesticide. That's why each time we miss already a couple of hours, they have to do cleaning. So we just want to clarify on the disinfection treatment.
Yeah, right. Yeah, because the current disinfectant, the chemicals that I mentioned just now, they all don't belong to the harsh chemicals like the chemicals that you mentioned to me before. Those the chemicals quite harsh, and then the residues you hardly to leave it away, just leave it like that. You have to wipe them off. So it's not like a pesticide. This is something like a very common chemicals on it. Yeah.
Okay, great. Thank you.
No, it's
okay. Okay, I have one question here from Michelle.
Yep.
So she's asking, is it advisable to do a swab test pre-sanitization and after that another swab test post-sanitization?
The sanitization here, Michelle means is a disinfection, right? Because
yeah,
to me,
I think so.
Sanitization to me is something like applied on the skin one. But doesn't mean I do. Okay, this is a very good question, Michelle. Thank you very much. I think okay, this is question. Usually, if let's say the area itself is considered as a endemic area, like found to be like a disease area. So usually all the PCO or whatever, they need to do all these swab tests. They are required. They are not required. They are recommended to do the swab test first to ensure that all the cleaners are safe after doing their disinfection. But if let's say some of them, they only do the work within the public area, but no COVID-19 is found over there, so the swab test is not recommended. It's not necessary at all. Yep.
Okay, I would like to add on this swab test because it's quite new to our industry. So when let's say a PCO want to carry out a swab test on the pre and post disinfection treatment, what are the parameters that the PCO has to know? Because when we go and talk to chemists will ask you, what are the parameters that you want us to test, you know? Dr. Leow?
Sorry, you mean I thought you are talking to the speaker. So can you repeat again? Sorry.
Okay, I would like to know, right, if let's say we proceed with the swab test for pre and post disinfection treatment, so what are the parameters that we need to inform the laboratory to test, the parameter of the swab test?
Oh, the parameter of the swab test, this is something because at the moment, the parameter here, you mean what kind of usually what they did is they will take the swab and then they go back to the clinic lab. They only will look into the, like recent one, they will look into the COVID-19 virus only.
Great.
So, okay, the parameter, they don't set a very complicated parameters at the moment. They're not going to go into other bacteria or viruses, but they're only going to want to know whether we are infected with the COVID-19. Yeah. So usually they will get the swab and put into their kit and to just like that, because the kit already itself is very specific to the COVID-19 virus. Yeah.
So that means the swab test, the entire point is we just have to make sure whether actually this is a before and after treatment. So the swab test, the whole entire objective is to see whether there's any presence of a COVID-19 out there or not. Am I right?
Correct. Yep.
Okay. If let's say the swab test, it's not necessary for us to do like bacterial count or things like that, because most of the time when we do swab tests, the chemists or microbiologists, they will straight away do a bacterial count, which in this case we don't really need to go into that. Am I right?
Yep. Not necessarily. Not at this point, because all these are the viruses. You cannot count it using normal way. We have to count using we call it real-time PCR in the lab quantification. So we have to use a molecular way, not the normal conventional way.
Okay. Great. Understand. Thank you.
Yeah, thank you. No worries.
Okay, okay, I don't see any more questions already. So I think we can close already, because we are already 15 minutes past our intended time. So let me just highlight upcoming training. So we have one training session that's still coming to everyone. So again, this training session is sponsored by Sequest Technology. And we have one final topic which is free for everyone. That's going to be happening next week on Thursday, 13th August. Quite a lot of you have signed up already, but registrations will be open all the way till next week. So look out for the email, which will be sent out to everyone on Monday with a reminder to be the link for everyone. So I think that's it for today. Thank you everyone for making time today and taking time out of your busy schedules to come in and listen and to learn. So it is now 11:46. Thank you so much again. Thank you to Dr. Leow for making time as well to share with us.
And thank you to UBE. Thank you to everyone. Nice to see you all.
Okay. So thank you everyone. So today we'll close the session today, and hope to see all of you next week for the next training session, which is an introduction to process NPD. All right. Thank you everyone.
Thank you. Cheers.